principles of the human brain. Following that, I'll explain how advances in neuroscience methods are allowing scientists to ask new questions about how the brain works. The goal is to set up the next few lectures on the neural mechanisms for language on the moment-to-moment time frame. Before I present the three big principles, let me briefly start with a basic assumption about the brain. You can think of this as a fundamental axiom of cognitive neuroscience. Recall back to the first lecture when we discussed the dangers of reductionism. Even though most scientists believe that the mind can't be reduced to only what happens in the brain, all scientists do agree that the brain is involved in every aspect of the mind. That's just basic biology. Denying this would be like saying that heart cells aren't involved in every aspect of blood circulation. Sure, they may be more or less important for different parts of blood flow, but without them, the system just doesn't work. Same with the brain and mind. So to understand how someone could believe that a loved one was replaced by an imposter, looking for clues in the brain is a must. The first principle is neural specialization. During prenatal development, genes help to direct traffic, guiding which cells should migrate to which parts of the brain. Most of these cells are designed to go to very specific places and to serve highly specialized functions. And once they arrive, many require further input from the environment to activate their particular roles. Once a cell's function is set, it usually serves a very specified and stable function over an individual's lifetime. For example, consider vision cells in the occipital lobe. Cells in this area have very specialized roles, and they're organized in a highly structured way. There are different cells dedicated for processing color, edges, and motion, and each of these cell types is located in its own neighborhood in the visual system. This pervasive localization and specialization is called topographical organization, and it exists not just in vision, but in all sensory systems in the brain. So let's connect this to our Capgras example. When a Capgras patient views her father as an imposter, there's a very specific part of the brain that responds to the face of her father. And you already know the name of this region from an earlier lecture. It's the fusiform face area or expertise area. The FFA is on the outer edges of the occipital lobe. And as you know, it's specialized for processing highly meaningful visual stimuli, like faces. Although the exact location of these face-specific cells is different across people, they're in the same basic neighborhood for everyone. This is important because it shows two things. Although the brain shares the same general organization across the population, the specifics of that structure vary greatly across individuals. This principle of specialization works in tandem with the second principle, which is this: the brain is an elaborate network. Yes, some basic functions of the brain are localized in just one area, like visual cells for processing color, edges, and motion. But complex processes like vision must integrate these sub-processes, so an interconnected neural network is required. This means that for most aspects of the mind, you must understand how the specialized subregions of the brain interact. To illustrate this, the neuroscientist David Eagleman uses the metaphor of a city. Suppose you are interested in where something as complex as the economy is located in New York City. It doesn't make sense to pick out a particular address or even neighborhood and say, there, that's where the economy is. Instead, you need to think about it as something emerging out of the interaction of many different locations in New York. Running with this metaphor a bit further, it's worth pointing out that certain neighborhoods of a city have unique features, and they even serve specialized functions, like districts for finance, entertainment, and housing. And for New York to function properly, these different areas must be connected by things like sidewalks, roads, and bridges, not to mention pipes, wires, and satellite links. This metaphor highlights a crucial point about the brain as a network. The specialized regions of the brain are no more important than the connections that bind them. Breakdowns in either of these things cause major problems for the brain. We've already seen this sort of breakdown with prosopagnosia. Remember, that's when people can't recognize faces, even their own. That disorder is caused by damage directly to face processing cells in the FFA, but the connections to other areas are fine. Capgras delusion is the opposite. The FFA is undamaged. It's the connections that are broken. The exact mechanism involved in Capgras delusion is still being worked out, but we know from research on brain damage that it involves links between the FFA and other key brain areas. One mechanism favored by V.S. Ramachandran is that connections between the FFA and emotional hubs in the limbic system, they're disrupted, and this causes people to view faces of loved ones without that old familiar glow. So a Capgras patient would see her father but feel nothing. Another mechanism put forth by Michael Fox's team at Harvard Medical School implicates connections with the right frontal lobe, which is involved in reasoning about one's beliefs and keeping those beliefs grounded in reality. Putting these two mechanisms together, it's possible that Capgras patients are alarmed that they don't feel the right emotion when they view a particular loved one. And a disconnection to the frontal lobe allows the brain to spin an unbelievable story to explain why. Incidentally, the brain is a wonderful storyteller. More on that in later lectures. This network property of the brain is so important that the National Institutes of Health have an official name for it, the connectome. We've made great progress mapping the location of specialized brain regions, and the NIH is heavily invested in the next frontier of charting the connections among these regions. The expected payoff is not only to advance basic knowledge about how the mind works, but also to help treat diseases of the brain like Alzheimer's and depression. Now let's turn to the third principle, and it's one that we've already discussed at length. The brain is plastic and built for change. This is especially true early in life, but even though it gets less plastic the older you get, your brain is capable of change throughout your lifetime. It had better be, or else you'd be in big trouble. Every time you learn something new, no matter how old you are, your brain changes. At the biological level, neural change is what learning is. This plasticity is integral to the first two principles. Even though genetics sets the basic blueprints for the brain, experience with the outside world is necessary to activate and fill in the details for many functions. For example, this is true in the FFA, where experience with particular faces is necessary for specializing cells to process those faces. Think about it. Nobody is born with specific cells in the FFA that innately fire in response to her father. Those cells must be first specialized through a meaningful visual contact with a person's father after she is born. Same story with the connectome. These brain networks are not fully in place at birth, and they require extensive experience to connect up all the parts. For example, at the same time the FFA is specializing for faces, it's also making connections to brain regions that give meaning to those faces. You know that familiar glow that comes from viewing the face of a loved one? That's not built into the FFA. It comes from connecting the FFA to the limbic system, which produces a learned emotional response to the face. This connection builds up through Hebbian learning, which over time strengthens the links between the two areas. If this connection gets damaged, it can cause major problems with face processing, even though the FFA and the limbic system are perfectly fine. Plasticity is also the key to recovery from broken connections in the brain. There are no drugs to fix brain damage. The main treatment is to undergo intense mental rehab. This seems obvious in cases of most other physical injuries, like a torn ACL, where extensive rehab is necessary for recovery. The same principle applies to recovering mental functions. What do you think clinical psychologists are doing for patients suffering from psychological problems like depression or obsessive-compulsive disorder? They're trying to rewire the connections of the brain to change how the patient experiences the world. So if a patient suffers from Capgras delusion, one treatment may be to repeatedly associate pictures of a loved one with positive emotions. Over time, this may allow weakened connections to get stronger, And in highly plastic young brains, it could even help create new connections to replace the old ones. So there they are, the three basic principles. The devil's in the details, but these general guidelines will help make sense of the next few lectures. What I want to do now is to give you a little bit more background on the techniques that neuroscientists use to measure and test these basic brain principles. So far, I've kept things pretty superficial with regard to neural methods, but I think we're ready to dive in a little deeper. According to President George Herbert Walker Bush and the National Institutes of Health, the 1990s were the decade of the brain. Looking back, it really was an exciting time for neuroscience. There were so many areas of growth, but one of the most significant legacies was the spread of neuroimaging techniques available to scientists. In general, these techniques investigate two basic things. There are spatial tools for helping researchers pinpoint where things happen in the brain, and there are temporal tools for recording when they happen. This spatial and temporal information provides two pieces of the puzzle in helping researchers know what is happening in the brain. Think of each type of information as a clue that brain detectives can use to solve a mystery. Imagine a crime where a museum discovers that a precious painting has been swapped with a forgery while being transported from one city to another. For any detective on the case, one of the first orders of business is to find the crime scene. Where did the switch happen? Now suppose there's a grainy surveillance video that identifies two possible suspects switching the painting just when it arrives to the new museum. That's the location. The next key step is to determine whether the suspects were in the vicinity of the crime scene at that time. If you could prove that they were there at the exact time of the crime, that'd be a lot better than proving that they were there